to the science group of the BRLSI, and I'd like to begin very briefly by reminding you to stay mute. And really a good idea to turn off your cameras, please. So stay muted and camera off. We've actually got a large audience tonight, and we could conceivably have bandwidth problems if you don't stay muted and camera off. We will have a question and answer session at the end of the talk, and at that point I'll revise those instructions, so we'll talk about that then. Okay, so I'm Stuart Reynolds, and I'm one of the co-conveners of the science group of BRLSI. And tonight, I'm going to have a conversation about the science behind the COVID-19 pandemic with two of my colleagues from Bath University, Ed Feil and Andy Preston. And our idea is to dig down and get a better scientific appreciation of why we are where we are now and what the future may hold. And what we want to do is to give you an overview of the general scientific consensus. Science is not unlike other human activities in that sometimes we disagree among ourselves, but scientists are very cautious, and there are widely accepted ways of moving forward to agree on what is the most probable explanation of what we see in the world around us. So scientific consensus emerges, and it's not our purpose tonight to discuss the political aspects of the response to the pandemic, although at times it may just be hard to avoid mentioning it. Edward Feil is Professor of Microbial Evolution in the Milner Centre for Evolution at the University of Bath, and his special field of research is in analysing the genomes of pathogens to understand how they evolve. And his work has had a big impact on tracing how infections spread within host populations and how resistance to therapies evolves. So, good evening, Ed. Are you there?
Good evening, Stuart. Yes, I'm here, ready and willing.
Terrific. And Andrew Preston is reader in microbiology at Bath, and his speciality is in the infection biology of pathogenic microorganisms, with an emphasis on understanding how human pathogenic microbes emerge from environmental reservoirs. Good evening, Andy. You there?
Good evening. Good evening, everyone else. Pleasure to be here. Thank you, Stuart.
Great. So, just to set the scene, I'm going to say a few words about where we are now, or where I think we are now, and where we've come from. So, late in December in 2019, just over a year ago, a new disease was first recognized when in Wuhan province in China, people began to experience respiratory symptoms and subsequently developed severe pneumonia. And some of these patients died. And the disease was dubbed COVID-19 because the causative agent was quickly recognized as a previously unknown type of a well-known class of viruses, the coronaviruses. And it was quickly recognized that this disease spreads very readily and had serious effects. And so strenuous efforts were made to contain it by restricting people's movements. But unfortunately, these didn't prevent it moving very quickly beyond the borders of China, and the disease was declared a pandemic by the WHO in early March. And at the same time, scientific progress in characterizing the virus and developing testing techniques was outstandingly rapid. So, for example, the virus's RNA genome was sequenced and made publicly available in just a matter of days. Today's figures from the WHO indicate that worldwide there have now been about 100 million people who have tested positive for this virus, and about 2 million of them have died. Those figures aren't very exact, as you'd expect for global figures. They're actually almost certainly a big underestimate, particularly of the number of infections. Here in the UK, the latest government figures, and I checked just yesterday, show that 3.7 million confirmed cases have now occurred. But recent data from random sampling of the population indicates that this is actually a gross underestimate. That discrepancy is because about a third of the people infected show no symptoms at all, and therefore that these cases wouldn't have been detected because those affected people wouldn't have complained and gone to the clinic. They didn't know that they had the virus. The probable real number of infections is estimated to be about 5.4 million people. That's 10% of the adult population. And that percentage is still increasing. As you'll know, because a lot of fuss has been made about it very recently, the cumulative number of UK deaths yesterday exceeded 100,000. Sorry, it's the day before yesterday. It exceeded 100,000 people. It's a very grim milestone. And taking into account the symptomless cases, this corresponds to about 1.85% of those who are knowingly or unknowingly infected who go on to die. That's actually quite a bit higher than the world average of 1.37% of deaths among the total number of infected people, if we allow a similar proportion of symptomless cases occurring everywhere. And this means that the UK death rate is rather high. I don't think that we should place too much emphasis on the exact numbers here, but the point to take home is that the UK figure of deaths of those who are infected is quite high, and there's been rather a lot of scientific discussion about why this would be the case. So that's where we are now, and let's start there as we begin our conversation. So, Andy, the president of Brazil once famously described COVID-19 as just a little flu. It's turned out to be a lot more than that. In fact, I think that over the past year, people everywhere have actually grown more alarmed at the severity of this new disease. Do we understand the biomedical reasons for why COVID-19 is such a medical problem?
Well, indeed, I think, some of those early comparisons to flu were perhaps inevitable, but actually pretty unhelpful at the start. There's some, significant differences, obviously, now between COVID and flu. So the high sort of medical consequences of this really come down to two combining factors, one of which is the actual sheer number of infections that there's been globally for this. And then secondly, it's the clinical consequences of those high numbers of infections combined to give the very, tragic figures that we're seeing, obviously within the UK, but globally too. So in terms of the numbers of infections, unlike influenza, the human population was pretty much entirely naive to the SARS-CoV-2 virus. So that meant, we'd not experienced anything closely related enough to this virus to give any type of pre-existing immunity. So we have the entire world's population highly susceptible to infection by this virus. And that's very different from influenza again, because of course we've been living with that for probably thousands of years, and we get residual cross-reactivity, memory from previous infections that actually dampens the effect of influenza every single year. So we've got a highly susceptible global population and a highly infectious virus.
So this is a bit like Columbus getting to the New World and importing a new disease that no one's ever seen before.
And indeed, and we do see it sporadically with influenza. If we get a sort of new assortment of one of these terrible avian influenzas that's sufficiently different that humans don't have large-scale immunity, of course, we fear that it rips through. And that's sort of, as soon as COVID was announced to be a pandemic, I think that's when the flu comparisons, because obviously we've been gearing up for another flu pandemic for a number of years, that's when they really took hold. Of course, you know, a respiratory spread virus is perhaps the easiest to catch. It's passed between people by the very act of breathing, which of course makes it particularly difficult to avoid that behaviour. Large, you've mentioned, the large degree, again, unlike influenza, I think, that asymptomatic infection, although it spares a good number of people who do get it from disease, actually makes it far more difficult to control because, of course, you know, if we're relying on people displaying symptoms to identify them and for them to take precautions such as isolation or seek treatment, and of course it's very difficult for, you know, in the animal world we tend to avoid other clearly sick members of our cohorts as a protection mechanism. We can't do that with asymptomatics. So of course we could be stood next to somebody in the supermarket, on public transport, completely unaware, no signs at all that they might be putting out the virus. So asymptomatic infection has been a real confounder in terms of the spread and persistence of this virus within our populations. Very difficult to do with. And then the actual virulence of the virus itself is sufficient that enough people succumb to disease, and unfortunately enough come to severe disease, that it of course places enormous strains on our medical facilities, and of course actually then starts to accumulate these terrible figures. So that those two combined, numbers of infections, ease of spread, and then it causes enough complications in enough people for it to really become the serious problem that it is now. And those, you know, for most people, they'll get very mild disease, if not asymptomatic. But again, you know, not unlike a number of other infections, in some people with you know, mainly with predispositions, it can trigger some sequelae, which are typically involving over-inflammation, so an dysregulated inflammatory response that sets off conditions, particularly involving the circulatory system, the cardiac system, in addition to the respiratory complications associated with the respiratory infection, that leads to actually most of the deaths associated with this disease. And that happens.
So this is like collateral damage from your own immune system.
Indeed, and of course, you know, there's many diseases where that happens. So sepsis, one of the big killers in the world, usually triggered by an infection, but it's not actually the infection that kills us in sepsis, and COVID-19 is no exception. It's actually, you know, it triggers in some people a dysregulated response, and then the body's inflammatory response that's, you know, in most cases protective, becomes pathogenic in of itself. And so you hear the term the cytokine storm. It does terrible damage to, for example, our blood vessels and the muscles around the heart. And most people, unfortunately, will die of either cardiac arrest or indeed multi-organ failure, as the rippled effects that start off in the lungs, without doubt, go across the entire body system.
Right, so cytokines are immune messengers that recruit these defences, and if you overdo it, it's bad news. Indeed,
they have direct effects themselves, but of course they're basically stop and go signals for the other parts of the immune system and effects on our other bodily systems. And it appears, you know, in some people, unfortunately, they get a sort of ongoing go signal, but none of the stop signal, and recruits more and more cells to the parts where these cytokines are being released. And the immune cells themselves can actually damage our systems. They have released molecules that damage the very fabric of some, for example, our blood vessels. And the other thing is that inflammation involves fluid accumulation. And so severe inflammation in the lung causes fluid to move out of the blood vessels into the lungs. That's where you get fluid accumulation, or this edema or pneumonia in the lungs. And of course, that can prevent the gas exchange, so the intake of oxygen, the output of carbon dioxide, that's the critical function of gas exchange or breathing in the lungs. And so you get a loss of respiratory function. That's the ARDS, the acute respiratory disease syndrome. and of course these other more systemic involvements associated with that very acute inflammation.
Right, so that's why they want us to get these finger oximeters so that we can look out for reduced lung function.
And it's also the case that it happens very, very suddenly. And so long before people really succumb to it, you get this very rapid, sudden drop in the oxygen levels in the blood. Actually, people can still feel quite normal at that point, but it's an early indicator that they're about to go downhill clinically very, very rapidly.
Yeah. Okay, just before we move on to thinking about infection, there's a last point that I find quite interesting here, which is that it does appear that not everybody is equally susceptible to this disease. A lot of people experience this as a very mild infection, and other people are hit for 6. And we hear a lot in the newspapers about how this is all about social disadvantage and underlying ill health. And I've no doubt at all that both of those things are true. But it does appear that there may be some genetic predispositions to being badly affected.
Indeed, I mean, we'll talk about some of the comorbidities perhaps in a few minutes. But you're right, and that applies to other infectious diseases as well. So any genetic predisposition that you have to perhaps a heightened inflammatory response is going to put you at risk from severe COVID. There's been talk about other genetic predispositions to the actual infection themselves. I think that data is a lot less secure at the moment, but certainly it's, we can look at those cases that go severe, look at those cases that are able to seem to shrug it off much more easily, and that sort of heightened inflammatory response, or possibly actually starting off from a heightened inflammatory sort of ground state, is a clear sort of predisposition towards that much more serious manifestation of disease.
Yeah, it's an interesting thought that it might ultimately be possible to identify genes in the general population that predispose to a bad outcome, so that such people can be better defended in the first place. It's a very interesting area, I think. Okay, let's move on a bit. And Ed, I'd quite like to recruit you here. During the last month, the number of new daily cases per head of the population in the UK has been very high compared with the rest of Europe. And I'm going to share my screen now and show a couple of graphs. Now, here we go. I hope everybody can see that. So, can you see those graphs okay?
Yeah, we can see them.
Right.
Yeah.
Okay. So, those two graphs show how the rate of confirmed COVID case deaths, which is the graph on the right, have varied during the pandemic in the UK and in the rest of Europe. And for our audience this evening, I should just mention that these graphs are from a really terrific website called Our World in Data. It's a very reputable and accurate source of data, which I can strongly recommend. And you can easily find their website just by Googling Our World in Data. But just looking at those graphs, what both of those graphs show is that in the spring, there was a wave of cases that then declined again in May. So a peak in April. So that's over here with infections and over here with deaths. And actually comparing the two graphs shows that the case rate data was probably severely underestimated in both the UK and in Europe, because testing at that time wasn't easily available, and we can guess too that the case rate was more severely underestimated in the UK than in Europe, because the death rate in the UK, which is shown in a sort of red colour here, is much worse than in Europe, whereas that doesn't really show up on case numbers. Why that high death rate in the spring happened is complicated, but undoubtedly the slow response of the UK government was a factor, as was poor handling of cases in care homes. But the point that I want to make here is that interesting things start to happen after our summer holiday. So you've got this surge of cases in the spring. We got that little bit of respite during the summer, and then around about the beginning of September, interesting things started to happen. And maybe you just talk us through what's going on here, Ed.
Sure, Stuart. So many people predicted that there would be a second wave come September, October. This is a common feature of lots of respiratory diseases and comes from multiple factors. One of which is that we know now pretty clearly that transmission is much more efficient indoors than outdoors. When the weather starts to turn, we spend more time indoors, which can also help, of course, explain the big dip during the summer months. Schools were going back, universities were opening again. We have a lot of a mass movement of people from those aspects, as well as the colder weather. The measures that were put in place weren't very effective in the UK very early on during October. So we had, if you think back, it seems like a long time ago, we had various sort of iterations of the rule of six, and then the tier system, and none of these really were quite enough to actually nip this surge in the bud. We didn't build up our test and trace capacity during the summer months, which was frustrating. That would have helped also to mitigate this surge. And of course, the how well that works is dependent on how many cases there are. There reaches a point where really, when there's too many cases to manage, then it has a much more negligible effect than if the number of cases are very low. So really, as in the first wave, we kind of just let the cat out the bag, so to speak. There was, as the tier system came in, the tier 4 system came in, you can see that there was a leveling off of
so that's death. Here, isn't it?
The dip in the cases, yeah. And it looked promising for a long time. And I remember sitting in, so Independent Sage do a really good video conference update every Friday afternoon. And I sat in on one at it must have been sort of mid-November, about that dip, when Christine Pagel pointed out that there was actually a worrying sign in the data that the number of cases had started to increase in London, particularly, before the tier 4 lockdown had ended. So even whilst we're in this still relatively tough level of restrictions, the number of cases did actually start to increase in London and the southeast, and we know that there was a big surge of cases, particularly in Kent as well. And it now seems likely, almost certain, that a lot of that surge of cases was down to something that no one had predicted. People thought was theoretically possible, but no one had really predicted came as a bit of a surprise, that there was this emergence of this new strain, which did seem to be transmitting much more quickly. And I think that all these factors combined together, not just the emergence of this new strain, but all the other things I've talked about, will feed into the big surge in cases that we saw.
Yeah, so here we are then. I don't know if everybody can see my pointer here, but here we are in November, and actually the British response here has not been so bad. I mean, there's a lot of hair, shirt and breast beating about how bad we are here in the UK, but actually the figures are not really so different from what's going on in Europe in terms of the number of cases per million people or the number of deaths per million people, right up until mid-November. And then bang, all of a sudden, the case rate goes wild here, and so does the death rate. And this is what's happening just after Christmas. So that doesn't look like a typical autumn seasonal wave, does it? Something different's happening. Andy, I'm going to share a different graph now, if I can just find it. Where are we? Here we go. So what's happening here, Andy? This is some data from the research group of Neil Ferguson at Imperial College in London. And what's he doing? What's he measuring here?
So as Ed mentioned, the new variant that I guess we all horribly familiar with now, we think arose in Kent, possibly sometime during September. I remember seeing some tweets early on with people saying, what on earth is happening in Medway, which seemed to be sort of the epicenter at that time, where its case per 100,000 suddenly skyrocketed. And everyone, what on earth is happening in Medway? And then, of course, it spread outwards through into London via the commuter belt and southeast. So this is basically a graph showing that this new variant, so the variant of concern, as PHE called it, which I think is a brilliantly apt name for these things, how its frequency has really increased exponentially during particularly November. So at a time where we would expect sort of everything to plateau because, again, of that lockdown in November, as Ed alluded to, this variant was actually still transmitting and seemingly growing at a rate much faster than the previous or the currently other circulating viruses at that time. So this was really an indication that the dynamics of infection had changed and we had something new had arrived on the scene.
Yeah, Ferguson is a very smart guy, and here he seems to be using two different sets of methodology to track the increase in the proportion of this new variant in the infections that he's measuring in London, the east of England, and the southeast. So it's actually in several different places at once, and it's tracking all together. But actually, I've got another graph here, which shows that actually not everything is all the same altogether. This is some data that just came out just the other day. You drew this to my attention, Andy. So this is from the Office of National Statistics. Do you want to take us through this, Andy? What's going on with the blue lines and the?
So we've had these sort of unbiased surveys running really right the way back from early May. So rather than taking everything on the basis of cases, we now have these population level surveys where swab tests get sent out to randomly pick people. Ipsos Mori are involved with choosing these to try and get a handle on what's happening kind of across the population. So different regions of the nations, the 4 nations, different age cohorts, different occupations. And people just, get sent some of these swabs. They take their own swab and send it back. And so we get this picture of positivity. So, every period where these surveys are conducted, there's maybe 110 to 130,000 samples taken. And essentially ONS just measure the positivity. So the numbers of those swabs come back that are positive. And these graphs actually split those positive swabs into those that we think are due to the new variant, shown in blue, versus those that are due to some other variants. And it's a nuance of that new variant that means it can be distinguished on the basis of those PCR tests, which is fortuitous, I guess. And it shows that, I think the rhetoric over the last couple of months has been that the new variant has driven absolutely everything associated with the current crisis that we're in. But actually the picture is a lot more nuanced. And it clearly shows that this was a sort of southeast phenomenon. And it really did start to dominate the picture of infection in those areas. But it's not been quite the same right the way across the country. And, I think, again, I think it's been portrayed that perhaps we're now at a point where only the new variant is causing COVID across the 4 nations, but that's clearly not the case. It's been significant in driving it, but it's not uniform. And I think some of these, again, this is a snapshot. So this was just the week, basically sort of the last 10 days or so. And I think we're seeing a sort of time effect that you get this delay as it moves across the country and it gets into an area, starts to dominate and perhaps even starts to decrease, as we're seeing in those top 3 with London, east of England, the southeast, whereas others may still yet to feel the full brunt of this. Now, whether we can dampen the effect of the new variant in other areas because of the current lockdown restrictions, time will tell, I guess, on this. But it's clearly a lot more nuanced than one variant coming through and causing all of the disease that we're seeing at the moment. There's complex sort of coexistence between the new variants and other variants in different parts of the country.
Yeah, so this is the southwest down here. We're obviously pretty interested in that here in Bath, and actually the new variant, the UK variant, has not really taken off in the same way here at all.
No, I mean, it has still increased because, again, you'll note that the other ones have remained relatively flat. And it's interesting, of course, I think in the southwest in particular, the second wave has really been the first wave. We got off very lightly in terms of total numbers of cases, numbers of people in hospitals throughout sort of the early spring and summer. And it's only really come to real bare its teeth this autumn. And actually, it suggests that we haven't been so affected by this new variant. So the real rise in cases that we've seen, sure, that new variant has contributed some of that, but there's also just been this sort of seasonal effect of the return of the virus through to some of those factors that Ed highlighted a moment ago.
Yeah, it's a reminder that actually for every occasion when you think that you understand what's going on, it immediately turns out you don't. I mean, we've heard an awful lot about how bad things are in Wales, for example, but you can see here that the problem in Wales is really a problem with the original variant, and the new B117 variant really isn't playing much of a part there. So okay, so maybe we don't really understand quite what's going on there. Okay, so I'm going to stop sharing that one. Right. So that raises an interesting point, doesn't it, Ed? So we've got this new mutant virus here in the UK, and we've just been discussing that it's at least partly responsible for what's going on. But maybe not entirely. But let's just backtrack a bit and think about how did that happen. Coronaviruses are actually not well known for their rapid rate of mutation, are they? Let's put up another picture here, and maybe you can. This is a pretty complicated diagram, Ed. You chose this. I think you better explain it to us.
It's very tricky.
What's going on?
So what's going on here? This big black line going upwards essentially illustrates the increase in the number of mutations as they accumulate over the last year. So from the start, we start at January 2020, the origin, then we go all the way through to January 21. Now each dot represents a sequence genome of the virus.
That's a lot of genomes.
This is hardly any genome. So here we've got 2,490 genomes showing on this graph. These are all from Europe. This is a sample from, I think the total number of genome sequenced now is about 270,000. So a really astonishing sequencing effort, about half of which have been sequenced in the UK. So it's one thing that we are genuinely well beating at, actually, I have to say. So what this graph depicts is the accumulation of mutations. So if we start from the beginning of the pandemic, back in the beginning of 2020, there was the first genome sequence, the Wuhan reference genome. As we carried on, as the pandemic progressed and the virus spread, so more and more sequences were generated. And these were all compared then to that original reference genome. So this in itself illustrates an important point, that there was one single point of entry. There was one original genome that went into the human population. Okay, so
the further up the graph goes, the more
mutations there are. So you have time going along the bottom, and you have the number of mutations going up the side. So as time progresses over the year, we get to more and more recently sampled sequences, so they have picked up more mutations compared to the original reference sequence from Wuhan in China.
So this dark line, the slope of that line, represents the mutation rate.
Exactly. And you can see, even though there's quite a lot of scatter around that line, this is to be expected, because mutation is, after all, a random process. It happens in a clock-like way, but there's no a definitive way of saying exactly when a mutation in a particular genome is going to happen. So there will be scatter around this line, but actually the line itself is very robust, and it gives us a very clear idea of how fast the virus is mutating. And you can see there's a rate estimate there, 23.058 subs, that's short for substitutions, is another word for mutations in this case, per year. So in other words, about one mutation every 2 weeks. And that calculation was made fairly early on, actually, in March, April, and it hasn't really changed since the days when we had just a few hundred genomes to now when we've got hundreds of thousands of genomes. So that's a robust calculation, and that's how we know that the mutation rate in this coronavirus, this RNA-based coronavirus, is actually at least twofold lower than the flu virus. So we're quite right to say that this virus is relatively stable compared to other viruses. But of course, there's been a lot of viruses, and there's been a lot of time for those viruses to accumulate mutations. So if you consider the 100 million plus people that have been infected across the world, each with billions of virus particles, each replicating within the host, then there is ample opportunity for just about every single possible mutation in this genome to have existed at some point, the vast majority of which we're never going to see, we're never going to sample or sequence, and the vast majority of which will just fizzle out naturally anyway, so we'd never see them even if we could sequence everything. So this is a tiny proportion. All these mutations are actually a tiny proportion of all the possible mutations that have existed out there.
Right. So do we have any ideas about how these mutations might affect the virus's ability to spread and cause disease? Do the locations of the mutations give us a clue?
So let's go back to what we were talking about, the UK variant of concern.
Yeah.
So if you could put your cursor onto that funny bit of the curve, that funny bit of the graph where it suddenly jumps above that line showing the accumulation of mutations. So that is those dots in that cluster there. They are our new variant B1117, the UK variant, and it's sitting well above all the other dots. And the reason is that it didn't just pick up one mutation when it emerged; it actually picked up 23, seemingly very rapidly.
So that's why it's standing above the rest of this line. Right. It's a step change.
It's a step change. It's an evolutionary jump. And this is really remarkable. We hadn't seen this before on all the genome sequences that we had. As I say, this is just the European ones, but if we had all the global collection, this would still stand out as unusual. And so we have to come up with a hypothesis what's actually happened there. What's interesting is we don't have any intermediate forms, so it's like a sudden evolutionary jump, and there's missing links. There's nothing linking the sort of relatively pedestrian rate of mutations in the predecessors to this new variant, which has suddenly accumulated all these mutations. So when this new variant was originally described, this was obviously demanded an explanation, and the explanation which those authors offered at the time, which I still think is the most plausible, and frankly it's the only one that's still been offered, is that actually what happened is that this variant, whether this was in the UK or elsewhere, we can't be certain, but the idea is that this new variant emerged within a single individual who was chronically infected with the coronavirus. So in other words, they're infected over a long period of time, maybe months. They were immunocompromised, so they couldn't fight off the virus. And instead they were being treated with a convalescent plasma. So this is plasma from infected people, a previously infected person, which contains the antibodies. So this is one way by which you can treat such patients. Now, if you expose the virus to these therapeutic antibodies in a single individual over a relatively long period of time, you're going to exert extraordinarily strong selection pressures on that virus to evolve, more strong than normal way a virus will be in a host for a relatively a short time, because all the selection happens within a host, because that's where the virus replicates, that's where the mutations emerge. So once this virus had emerged in this individual, this is all a theory, by the way, I should stress, then somehow it must have got out of that individual and infected other people, and then gone on to, after probably a period, a lag period of at least a few weeks, then started to spread more rapidly. The other thing that's remarkable about this new variant, and indeed the other variants as well, the Brazilian variant and the South African variant, is that many of the actual mutations we've seen before. They're not random new mutations. Many of them are mutations in this infamous spike protein, the gene that encodes for the spike protein, the thing that sticks on the outside of the cell. And this is really where the battle is being waged between our immune system and our therapeutic efforts through a vaccine, etc., against the virus, and where the virus is fighting back in terms of being able to bind to the host cells, etc. This is really the ground zero of the war, this particular protein. And many of those mutations, many of those 23 mutations actually occurred in that protein, and many of them are common to not only the other variants of concern, but also we've seen them before in other strains. But what's different is that we haven't seen them all in one virus before, all in one genome, all in this combination. And it seems likely that it's not just the effect of individual mutations, it's the effect of the combination of mutations. They can sort of work together and interact together in ways that we don't really understand, which potentially could increase how quickly they replicate inside a host, or how well they bind to host cells, and or how they evade the immune response. And all these things can have knock-on effects in terms of transmissibility. If they're better at living inside the host, if the host sheds more virus, then the virus itself becomes more contagious and spreads more quickly.
Yeah. As I understand it, the evidence that this new variant virus is more infectious, the experimental evidence is not that great. Really, the best evidence that it's more infectious is simply that it's replaced the other, the original kind of virus in the south of England. But then I guess that we have to be a bit cautious about thinking that that's really conclusive, because that's evidently not what's happening in Wales. But anyway, I guess that we shall see fairly soon. Yeah.
Oh, absolutely. And those experiments are difficult. I mean, the lab experiments that test the strength of binding to host cells and all these, and the rate of replication, and all these things are difficult experiments. They've obviously been ongoing since this variant first came to our attention in Public Health England and brought down in other places. But it's going to take weeks, if not months, before we have a definitive answer from the lab as to what these new variants might be doing. So the first line of evidence is the epidemic.
Ooh. Now, I think that we just lost Ed's voice there. I expect he'll be back in a minute. Meanwhile, maybe I can ask Andy. So the virus may or may not be more infectious. I think the balance of evidence is that it probably is. But is there really any evidence as to whether it's more virulent? Does it have worse or nasty effects on patients?
Well, again, this is the very recent suggestion that came from the recent press conference in Downing Street. So, but again, it's based not on direct laboratory or animal model studies. Again, it's looking at the proportion of people that enter hospital with the new variant compared to those that do not. Then that those that go on to need ICU support compared to those that have the old variants. Of course, unfortunately, those that then go on to die after infection with the new variant compared to those that do not. And 4 independent studies were reviewed recently, and all of them gave very similar sort of 30% increase in certain patients. Those parameters, so ICU requirements and unfortunately death, were all increased in patients that had the new variant. So again, there's quite possible there were confounders, so other factors that are hard to pull apart from it just being the new variant. Biologically, of course, it fits. You know, so you've got something that's spreading more. It's got mutations that we know affect its very, very likely affect its interaction with the host. Transmission is sometimes sort of the clinical consequences can go hand in hand. So it wouldn't surprise me if the data does turn out that yes, it is. But at the moment, I think it's hints from sort of clinical and hospitalization data rather than definitive sort of virulence studies.
Yeah, it's very difficult to do the kind of experiments that we as biologists would like to do, because of course they would be completely unethical. Yeah,
and it does take a while for the animal model experiments to come to fruition. So again, those are underway at Public Health England. But again, we're going to have to be careful making direct comparisons between, for example, macaques and humans. There are subtle differences in the way that we respond to this virus. So again, we may get supporting evidence, but not conclusive evidence coming out in the next few weeks.
Yeah, something that took a long time for me to realize as an infection biologist thinking about the evolution of disease is that actually it often doesn't make sense for a pathogen to cause serious disease. The pathogen's strategy is to reproduce as much as possible, and that might involve not making the host ill. And so I don't think that we can easily say that just because this variant is spreading well, that it'll make you more ill. The opposite might be the case. And we'll just have to wait and see. So, Ed, we lost you there for a moment, but I think that, yeah. So, but you know, listening to what Andy and I were just saying, do you think that we could ever have expected to be able to predict the emergence of new variants of the virus? And now that we know that they are emerging, do you think that there'll be more? I mean, are we in a predictive position here?
So generally speaking, there is no theoretical framework or general evolutionary law that would have allowed us to predict one way or the other whether this virus would become more or less virulent or more or less transmissible. Or clearly transmissibility is going to be a benefit to the virus, but what the consequence of that for virulence would be could go either way. So it's quite possible that virulence could actually increase transmissibility. It's not always the case that they follow this so-called law of decreasing virulence, that a virus will, or any sort of pathogen would inevitably evolve to become less virulent over time to get some sort of mutual understanding with the host. The real world is much more nuanced and messy than that, because there are multiple ways by which virulence and transmissibility can go hand in hand. For example, just being able to replicate quicker, most simply.
Yes.
So in terms of answering your question, generally, no, we can't predict which way it's going to go. And there are mutations, even in this variant, this UK variant, there is a mutation, a so-called nonsense mutation, which disrupts a gene called ORF8, which is basically means it's a gene that we don't know what it does. And this mutation, as many others in the UK variant, has been seen before in other strains. It popped up in Singapore, and there's a perfectly credible paper characterizing that mutation, suggesting that it actually decreased virulence. So mutations can have different effects either way, and then you have to have the combination of mutations, which again adds an extra layer of complication. Now that's the general answer. Where we are now, I think that there's actually it's quite telling that these three main variants of concern that we know about in UK, that are first sampled in UK, in Brazil and South Africa, they all seem to have happened more or less at once. Now, having said, described the theory for the emergence of the UK variant, the Brazilian variant in this, which emerged this was first observed in the city of Manaus in Amazonia. The idea there is that may have emerged due not to immunity accruing or antibody pressure in a single individual, but actually immunity in the whole population. So that particular city had a really, really high infection rate. It's estimated that 76% of the population of that city had previously been infected. And it's just possible that once that level of infection is high enough, that there's a pressure on the virus to somehow sidestep the immune pressure that's evolved in that population to try and skip around it. So there's an extra evolutionary pressure there for the virus to change. Again, that's just a complete theory, but if that's true, then that's a bit of a worry, because it means that all of a sudden we might see these variants cropping up in different parts of the world as the amount of immunity in the population hits a certain threshold. Now, I should stress that this is immunity by natural infection, which is quite different from immunity by the vaccine, and there's still no real reason to panic in terms of how these variants are going to respond to the vaccine. So that we'll probably come to that later.
Well, I was going to ask you that right away, actually.
Yeah.
You know, we're talking about the virus maybe being able to sidestep host immunity, but really all the vaccine does is to increase the immune selection pressure, isn't it? I mean, should we be expecting that varieties of virus will arise that are immune to the vaccine?
I think it's a possibility, but I think if it does happen, then it's not like an on-off switch. It's not going to make the vaccine not work anymore. It will lower the efficacy of the vaccine. And one of the really big advantages of these new RNA-based vaccine, like the Pfizer one, is that it's actually quite relatively straightforward to tweak the design to make it so that it's actually going to be effective against the new variant. And it's not the case that it'll have to go through all those phase 1, 2, 3 trials all over again. There will be some regulatory, not shortcuts, that's the wrong word, but it won't be such a long process to do that as developing a vaccine from scratch. So that's a great source of reassurance for me, at least, that even if it does happen, it's not going to make the vaccine not work completely, and it's not going to be the case that we can't modify the vaccine to in a sense, stay one step ahead.
So we could be coming to a position maybe next year and subsequently, a bit like the case with flu, where we're looking to have an adjustment of the vaccine each year to keep it up to date.
Yeah, I think that's perfectly likely. I mean, I couldn't put an odds on it, but I wouldn't be surprised if that happens. And we don't know how long immunity will last for anyway. We still don't know exactly what the T cells are going to do. We don't know how long, even if the virus doesn't change, we don't get new variants. We don't necessarily know how long the effectiveness will last for. We just have to, and there's no way of predicting that. We just have to wait and see. But yeah, I think it's certainly possible that we will have to have seasonal jabs like flu.
Right. Well, there you are. I'm due to get my first jab of the vaccine the day after tomorrow, so I'll keep you informed. Yeah, Andy. But by contrast with all this sort of muddiness about our understanding of the pandemic itself, the worldwide race to make a COVID-19 vaccine has been exceptionally successful, hasn't it? I mean, I can recall listening to pundits talking in April of 2020 saying, oh, well, if we're very lucky, we might have a vaccine by the end of the year. And here we are, and we have it. What have we learned from this great success? Is it entirely new thinking, or have we simply put existing knowledge to work?
I think we've sort of reaped the benefits of a decade of sort of real vaccine development. So, the Oxford vaccine, for example, that technology, so using those chimpanzee-derived viruses to deliver the S protein, that technology was developed just for this case, for this so-called disease X, a new emerging pandemic. And actually, just before we went into lockdown, the Oxford group published their phase 3 clinical trials into the MERS vaccine, so the Middle Eastern respiratory syndrome virus, created using the same approach. Of course, Moderna, they were actually a cancer vaccine company developing messenger RNA-based vaccines to try and develop immunity against cancers, so a sort of personalized medicine approach. And of course, it was recognized very, very quickly that it could be adapted to encode for anything. And so I think we were fortunate that we had a good heads up from SARS and MERS as to what the likely target for the COVID-19 virus was going to be, the S protein. And we had that existing sort of phase 1 and phase 2 data from using S protein based vaccines against SARS and MERS to know that they were going to be highly immunogenic. So there wasn't a long period of discussion as to what the vaccines were going to be based on. And then those two technologies were ready to go immediately. And actually that was really the sort of massive reduction in vaccine development that we've all heard so much about. And of course, if you've got almost limitless resources, the purse strings are suddenly opened, which is not the case for most vaccine developments. It shows what can be done. So I think, the one of the legacies from this will be hopefully really the demonstration of this new era of vaccine platforms.
So the approval of these vaccines has been exceptionally quick. Do you think that any corners were cut, or has it all been fair and above board?
No, I think we've had exactly the same evidence base that we would for any other new vaccine coming on board. I think one of the differences is that there was a very clear and immediate clinical need for implementation. Whereas in other sort of new vaccines, so if you remember the meningococcus type B one, there was a lot of discussion because actually the case rate had fallen away so much it took an appeal to get that vaccine put into implementation. Of course, the Ebola vaccines, they were ready to be given emergency use authorization, but just as the sort of the 2014 to 15 outbreak died away. Here, of course, it's been slightly fortuitous timing that they've arrived just as we're experiencing this horrible second wave. So a culmination of factors, but no, we do have to recognize we don't have long-term data on these vaccines. A lot of people are saying it's too early to use them, but then the question, sort of going, when would a good time to use them be? How long do we need to wait for long-term data? It's quite clear that most people, if they're going to have a bad reaction to a vaccine, it happens very, very early on. We do have 9 months of very careful monitoring of these trial participants. They bled more than most people would care to, so every facet of their physiology has been monitored, and we don't see any signals that cause concern. So, of course, we can't say what's going to happen 10 years down the line, but who would like to wait for 10 years before we try and come out of this pandemic? So no, the safety data has been the same as it would be at the same stage for any development of a new vaccine.
Well, those are encouraging words, and maybe with that we'll sort of stop the formal part of this discussion and throw the proceedings open to questions from the audience. So a general thing that I would have to say is that quite a lot of people have sent in questions by writing them on the chat button at the bottom of your Zoom screen, and I've got a number of questions here to work on, and I'll work my way through those. And if you want to add yours there, we'll get to you in the end. The other thing that you could do would be to press the reactions button at the bottom of your screen and wave a hand around. But actually, there are so many of you that probably writing your question would be a better one. And well, I think that I'll at least start off by actually asking people, particular people, to unmute themselves and activate their cameras so that we can all see you and hear you ask your question. And I'm going to begin with a question from Hugh Loxdale, Hugh, can you mute yourself and switch on your camera? Would you like to ask your question in person, please?
Okay. Well, I actually have three questions, but I just don't want to be greedy. But
you better not be greedy, Hugh. No, start off with the best question.
In the terrible 1918 flu pandemic, if you got the flu, this was often it was not fatal. It killed my grandfather in 2 days. But as we know from COVID-19, you have these asymptomatic and mild cases, and you also have the severe case. Is it possible that there are actually cryptic strains of the virus going in circulation, or we know it is definitely the same virus, except of course as you say, there's the new, very pathogenic strain. But the original strain was the. Do you think there were cryptic strains going around? Some mild, some asymptomatic, and some pathogenic.
Do you want to tackle that one, Ed?
Yeah, sure. So by far the biggest predictor of how serious the infection will be in terms of clinical outcome is age. So I think that probably when we're looking to explain the great differences in between the asymptomatic or the very mild cases and the very severe cases, then it's natural to look at the host more than the virus. Frankly, despite the fact that I've just shown that plot with all the mutations, accumulating over time, and stated that the virus has had a chance to sort of accumulate practically any possible mutation over time. The fact is that even now, when you compare the contemporary strains with the original reference genome, you're still only seeing 30, 40 mutations that have accumulated at most when you over that time, and this is in a genome of 30,000 bases. Mm-hmm. So actually, the number of mutations, the impact that the mutations have had on the genome is still relatively small, because it's really starting from nothing. So it's going to take a time. So I would be very surprised if there's really been time for the virus to sort of explore different strategies in the way you describe, something like sort of filling out different niches, I guess, if you're thinking about it ecologically. I certainly think that that's possible over the long term, but I don't think there's been enough time for that to happen.
Thank you.
Okay, I'm going to ask Chrissia Bell to unmute and switch on the camera. Chrissia, can you do that?
Yes. Can you see me? Oh, yeah.
Yes. Yeah, that's great. Could you please ask your question about tweaking non-RNA vaccines?
Yeah. I was rather alarmed to read that whereas the RNA vaccines could potentially be reduced in efficacy 6-fold, that the AstraZeneca or non-RNA vaccines might be reduced in efficacy by 10-fold, or its efficacy eliminated altogether. Given the UK's heavy reliance on AstraZeneca, we won't get political here, but the AstraZeneca vaccine, and the fact that it is not saving the RNA vaccines for the most vulnerable populations, it's not making a distinction. What's going to happen to all of the people who get the AstraZeneca vaccine if it turns out that these fears become reality?
Do you want to have a go at that, Andy?
Sure. So I think actually those figures are not a 6-fold reduction in efficacy. So I think this is the data that came out, forgive me if I'm wrong on this, but they showed a 6-fold reduction in recognition of the new variant by the neutralizing antibodies. But actually the level of recognition was still well above what we think would be protective. So the actual efficacy is the overall protection, so the percentage of people that would be, no longer getting COVID. So that efficacy, has not dropped sixfold or tenfold at all.
Okay.
We don't know the true effect, but biologically speaking, as Ed said, we might see a partial reduction, but certainly not anything like that. So I think that was actually a laboratory study that measured the recognition of the new variant by certain groups of antibodies. And although compared to the old variant, there was a statistically significant drop, the levels still remained well above which we would be hopeful to see a protective effect. But in terms of tweaking the vaccines, that's one of the great advantages of these new technologies. I mean, the Oxford group and Pfizer, they didn't even need to have the virus on site to make their vaccines. You take the genetic sequence, which of course we can generate pretty easily these days. So this is just looking at the code, so the RNA code for the S protein from the South Africa variant. You now replace the old variant code in the virus for the Oxford AstraZeneca vaccine with the new code, and you just simply switch production to that. So technologically, it's very easy. As Ed alluded to, we'd need to just have a little bit of safety data and a little bit of QC data. But I think we'll have by then good data on the type of immune response we would like to see. I think we can generate that within a matter of weeks of producing the prototypes for those new vaccines. So again, I think the head of Pfizer said we would take them 6 weeks to sort of be ready to go into production with a new tweaked version. And the Oxford vaccine is very similar in terms of how relatively easy it is to include the new variants in future vaccines.
And what about the people who've already received the old vaccine? Let's say if we manage to vaccinate 15 million people by the middle of February, and the tweaking happens in March, April, what will happen to those people who have been vaccinated with the old AstraZeneca, for example, or even Pfizer?
So I think these are not going to be such different that they would not, you're going to get a boosting effect from even if you're tweaked with one of the new variants, because again, as Ed mentioned, we're calling it a new variant. There's relatively few changes. So out of those maybe 8 or 9 mutations that affect the S protein, that's out of a total of many hundreds of amino acids residues. So although the changes may have a biological effect, the way our immune system will see them is it will still see very largely the same protein. And the way that the immune system mounts a response to it, our system chops up that protein into much smaller segments. And many of those small segments, which we actually end up reacting to, will be identical between the old variants and the new variants. So again, it's going to be the same boosting. It's just it will give you extra coverage against these new variants. And I suspect what will end up is in a couple of years' time, we'll all be having boosters that contain a cocktail of the different variants that are circulating at that time. They will still boost against the old variants, but they'll now provide additional coverage of whatever new variants come through. So you'll still see that overall increase in protection with a booster, even if it's with a slightly different variant, because they're still going to be so similar that you'll get some cross reaction between them.
So people could have to have 2 boosters, if you see what I mean. If they've already had the first 2 injections, first 2 inoculations, they might have to make a program where people are then asked to have another one this year, 3 in a go.
Whether it's this year or whether it's on a more yearly seasonal basis, we don't know how the virus is going to react. But absolutely, it is not, we have multiple boosters for some of our statutory childhood vaccinations over the first number of years of life. So we go in hard with say three hits, for example, with the pertussis vaccine, 2, 3, 4 months, and then we have a preschool booster just to top it up. So that we have no data to suggest what that booster schedule might look like. We'll have to wait and see, but it wouldn't be surprising that if this virus sort of continues to circulate amongst people, then boosters become part of our vaccine landscape for years to come.
Thank you.
Yeah, another interesting point about this is that, of course, a a vaccination protects you not only through your own personal immunity, but by suppressing the level of virus in the community. And that's something which isn't dependent on your own immune status. Okay, I've got another question here from David Hall. It's rather a tricky one. I'm not sure whether we'll be able to answer this. David, are you there?
Yeah, I am. I'm here. And first of all, thank you for your very clear explanations. It's much appreciated. So what is behind this question is really about vaccine take-up, which, of course, there's a lot of talk about that at the moment. And it's just a day or two ago, a doctor on the TV stated that the Pfizer vaccine should not be taken by those who have an allergic reaction to penicillin and ibuprofen, but the Oxford AstraZeneca vaccine is okay. So immediately you get a bunch of people saying, right, okay, the Oxford vaccine is safer than the Pfizer vaccine. I'm going to take that one, but I'm not going to take the Pfizer vaccine. So it is, first of all, it is correct, because it's the first I heard of it. I mean, okay, the layman in these issues, I noted that there was a rare allergic reaction to the polyethylene glycol, which is in the Pfizer vaccine. But it's first I heard of this. So is this correct? And if so, what are the reasons for it?
Can we tackle that one? It's a bit technical for us, I think.
Again, I don't think there's any official notification. So if you remember back very early on in the administration of the Pfizer vaccine, those two healthcare workers suffered that initial reaction. Again, they were known to have some really very severe allergy concerns. They carried EpiPens, but then I think it was worked back to the fact that it was, if you'd had a history of allergies to any vaccine, then you were advised to wait until the data. But actually, other medicines and food allergies were deemed to be safe. So I don't think that is correct. I don't think that's the official at the moment. Of course, what may happen is at the population level, you're always going to find somebody who reacts unexpectedly to a vaccine. So if that's happened in somebody and they happen to have a penicillin allergy, you're going to link the two. It doesn't mean actually the penicillin allergy was actually responsible to them, because again, they're entirely different compounds. There is nothing in these vaccines that looks remotely like a penicillin molecule that would be the thing that stimulates their allergy. So no, I don't, personally at the moment, I don't think that's sound advice, because again, there's no data associated with that, and there's no plausible biological explanation for why something that triggers it. For penicillin, we know the structure of that. There is nothing that looks like that in these vaccines. Yeah.
Now, this was my concern, that people would go on the TV in front of millions of people, might be overstating cases, so they should be very careful about what they say. We
will see a whole series of sporadic, perhaps, some potentially serious side reactions, and that's what happens when you scale up to what's going to be billions of people, I'm afraid. Yeah, we have to be prepared for that.
Okay, so I've got a question here from Trish Fosbury, although I'm not sure whether it's Trish or whether it might be Bob Fosbury.
It's actually Bob.
Hello. Would you like to switch your camera on, Trish or Bob?
Yes.
Hello.
Right. Hello. Yes, this is a question that's been going around for a long time without much resolution. It has been suggested, notably by David Davis, that this is actually a vitamin D deficiency epidemic resulting from slow adaptation of populations to different latitudes and environments and so on. There's yet few clinical trials to investigate this. There are some, but there are many, many examples of correlation data that support this idea. And in particular, I was struck by the statement by NICE, who chose one clinical trial to investigate this, where all the cofactors that they rejected as noise were cofactors that might have been related to vitamin D deficiency. Do you have opinions?
Can we tackle this one? I think we might be a bit out of our depth here. Don't know, Andy. You're probably the best qualified of the three of us to talk about this.
I would argue with the term that this is a vitamin D epidemic. This is a viral epidemic, and you don't get COVID without having the virus. So I think this comes down to the response to it. So vitamin D is one of these interesting things that does have multiple effects. So again, but it ties in. It certainly does tie in with the idea that those people that suffer badly from it have a dysregulation of their immune system. Vitamin D is known to have an inflammatory dampening effect. But again, I think what's happened is that because it's gained such popularity in terms of being a potential sort of panacea for staving off serious disease, there's a lot of studies have focused on the vitamin D levels in certain people. But again, if that's all you look for, then sometimes that is all you see. And actually, the one sort of real controlled trial that's underway, the initial data that came out of that, and again, it's one of those messy things. For some people, it might have some beneficial effect, but it's not going to be an absolute intervention. So we're in one of these messy things again. But that's the same with so many of these things. It's clear that not everybody who's over 75 who gets COVID is going to die from it. But we know a lot of, that it's certainly a risk factor. So I think the recommendation is generally from the BMA to ensure that you have good levels of all of your vitamins and minerals, including vitamin D. They have to be a little bit careful about it because it's one of the vitamins that you can overdose on because it is fat soluble, so you don't excrete it if you overdose, and that can have just as serious effect. So I suspect that when it comes out in the wash, it might help some people in terms of not going down with severe disease, but it's not going to be a uniform effect across the board because it has a number of then secondary effects on different aspects of the immune system.
And indeed, as quite a few people in this audience will know, actually, if you're considered vulnerable, you do have the offer of subsidised vitamin D from Mr. Johnson's government. Yeah.
And one thing is clear is that it's not going to do you any harm unless you overdose, as Andy says. So absolutely, as long as you don't think it's going to be a panacea and make you bulletproof and all the rest of it, it's not a bad idea at all to take it because it's good to have vitamin D. I mean,
it's probably an even better idea to go out for a walk in the open air.
Well, yeah.
Doesn't help you in the winter. We get no vitamin D from winter sun at all. So.
This is true. The light level is pretty low, isn't it? Yeah. Okay. Thanks very much, Bob Fosbury. Here's a tongue-in-cheek question from Trevor to everyone who says mutation seems to happen when the virus encounters a problem in reproduction. Does that mean the pathogen has intelligence? Well, I guess I'll answer that straight away by saying no. Are you there, Trevor?
I am indeed.
Yeah. Hello. Yeah.
My question really was about, is there any research to look at how we can prevent viruses and pathogens mutating?
That's a nice question, actually. Viruses are hardly alive at all, so that they don't tend to regulate what they do very much at all. But actually, there are some slightly more advanced pathogens that do indeed put their rate of mutation up and down according to the circumstances. Ed, you probably know all about this.
Yeah, for sure. I mean, there are bacteria which are incredibly advanced organisms compared to viruses, which are able to change the rate at which they mutate in response to conditions. So the mutation rate is actually an evolvable trait as much as anything else. There's a limit to how low it can go, but it can turn the dim switch to increase it. And what can happen in bacteria is if they find themselves in particularly stressful situations, conditions, they can throw all caution to the winds. This isn't I'm anthropomorphizing a lot here. This isn't a strategy that the bacteria works out. This is just what happens. The mutation rate a mutation can occur, which in turn will increase the rate at which other mutations happen. So that will be selected for. And if you increase that rate of mutation, maybe 100-fold, this happens in the lab very easily, then you're basically throwing caution to the wind and having lots of mutations in your genome and hoping that one of them is going to help you out of this hole that you're in, these stressful conditions. But I would say, coming back to the virus, so there are two things. Even though, of course, mutations are completely random, each individual mutation is a completely random event, there are theoretical things which we can do to limit the risk for mutations. Most simply, the more viruses, the more virus particles there are out there, the more humans that are infected, the more mutations there will be out there. The bigger the population you have, the more possible mutations will be existing at any given moment in time. But so by suppressing the overall size of the population, the number of viruses, that's one way by which you can decrease the total number of mutations that are out there. The other sort of train of thought is that I mentioned about the possible one theory about the emergence of the UK variant, that it was under this extraordinary selection pressure in a particular host undergoing this therapy with convalescent plasma. So if we can identify those sort of conditions, rather like the stressed out bacteria in the lab, that are likely to select for particular mutations as and when they emerge, then if we can avoid those sort of situations, then again, that's another way by which we can potentially mitigate the risk from these mutations really causing arising and then spreading.
Actually, Trevor, I thought your question was really great. And the idea that deliberately engaging in mutation might sometimes be a sensible response to an environmental challenge is actually one that has been taken up in a very big way by our own evolutionary lineage, because our adaptive immune system, which generates an almost limitless possible variety of immune responses, absolutely depends on mixing up bits of DNA in a rather mutation-like way. So, the essence of your question was, is it possible to take advantage of mutation to be adaptive? And the answer is yes. We are living proof of that. Right, I've got a very interesting... Oh, sorry, Trevor.
No, I was thinking about that in terms of our own immune system. And this, when I hear you people talk about it, it sounds like a battleground, and it is, of course, a battleground. And that's why I use the word intelligence there. That we could use our own immune system to really get into this battle and see where we can put, if you compare it with a military exercise, where best we can put our forces to battle against it. That was the point of using the word intelligence. But thank you for the answer. That was interesting.
Great. Thanks very much, Trevor, for that question. I've got another very interesting question here from Michael Perryman. Are you there, Michael?
Yes, I am, Stuart, but I don't have a camera. So
okay, well, we can imagine what you might look like from your voice. And would you like to tell us your question?
Thank you first for the three of you, the panel, for getting together and giving this overview of where we are on a more scientific basis. Very enjoyable. My question was really not much more than what I've written here, but would any of you care to speculate on the perhaps a little bit more detail what you think is going on with the World Health Organization investigation into the origins of this pandemic in Wuhan. Of course, it's in many people's minds, you know, whether this virus did escape from some research facility. But that element aside, you've discussed at length the idea of following these mutations as a function of time, but presumably what the World Health Organization investigation has to do is track it also in spatial terms, in physical space. And I was wondering, you know, if you could just give us any background at all as to what is going on and what you think might, what the prospects might be for this kind of investigation. Thank you.
Oh, this is a great question for Ed Feil, I think.
So it's difficult for me to speculate on this actual investigation, what's going to happen, because that's wrapped up in all sorts of geopolitical forces, which I can't even pretend to understand. In terms of the origin of the virus itself, it's not absolutely impossible that it escaped from a lab, but I do find it unlikely for a number of reasons. The first reason is that I don't see any reason to really go in that direction, to think in that way, because we know that out there in the wild, in nature, particularly in bat populations, there are hundreds, thousands of as yet uncharacterized viruses, much like this one. And in fact, this process of spilling over from the animal populations into man probably happens quite regularly. It's just that almost always the virus is so maladapted to humans that it won't be able to make that jump into onward transmission from one human to another. And for whatever reason, this virus did. So I don't think that there's any real necessity to think that this virus escaped from a lab. I also know a little bit about something about the safety procedures that happen in these laboratories. And let's be frank, it's great that these laboratories exist because we need to know about all these natural viruses, right? And you know, the safety procedures which are in place are really very, very restrictive in terms of working with live virus. The live viruses are locked deep in frozen vaults. They're not worked on hardly at all. People work on the dead viruses. It's kind of hard to imagine how an escape would have happened. I certainly don't think it was done deliberately. So I hope that's answered that part of the question. I don't actually know how the Chinese authorities how they're going to really respond to whether they're going to be as open as they are promising to be. I hope they are, but we shall see. In terms of the geospatial spread, well, that is absolutely a very good point, and that of course is the real raison d'etre behind all the sequencing, in that we've been able to track how the virus has moved from one country and one continent to another, and we've been able to identify when new variants or whatever what it is introduced into a region and spread within a particular region. We've been able to estimate how often, for example, the variant was the virus was introduced into the UK during those very important months of February and March last year, thanks to the genome sequence combined with other types of information. So that geospatial spread is really, to a large degree, the point of all the sequencing. The being able to measure the mutation rate is nice, and it's a nice illustration of how mutations work, and it's nice to have that very robust estimate of mutation rate, and that strange phenomenon of the new variant accumulating all those mutations at once. I mean, that really illustrates that point well. But the main sort of aims of all the sequencing is really to track the geospatial spread.
Well, I think that's probably enough now. We're a bit past 9 o'clock, and I think that probably quite a lot of people have had enough for today. We've spent the last year thinking almost continuously about coronavirus. So the idea that we might perhaps go off and have a cup of tea and stop thinking about coronavirus for at least half an hour or so seems quite attractive. I really hope that we've managed to tell you something that you didn't know, or perhaps that between the three of us we've managed to give you some perhaps rather better appreciation of the way that the scientific establishment thinks about this. And I think I could sum it up by saying that we're not out of the woods yet, but at least we can see a route through to where we might be in a better position. So I'd like, on your behalf, to thank Andrew Preston and Edward Feil for sharing their expertise with us tonight. And as a kind of a way of an experiment, I'm going to invite you all to unmute yourselves all at once and to clap. And let's see if that actually completely destroys the bandwidth.
Thank you.
Thanks very much, everybody, and I really hope that you've enjoyed yourselves. Consider joining the BRLSI if you're not already a member, and if you're already a member of the BRLSI Science Group, I'll be writing to you quite soon to tell you about our next meeting. So good night.